has this happen. So that would suggest potentially that there's some family history of swelling or edema. So just kind of putting these pieces together, we're probably dealing with a patient that has hereditary angioedema. A lot of times in board questions, they'll present this more with like lip swelling or facial swelling, but it can happen in the extremities. It can happen in the bowel too. And in this case, this is probably what's contributing to her severe left lower quadrant abdominal pain. So she was already kind of predisposed to this angioedema, and then you gave the patient lisinopril, and that kind of tipped her over the edge. So let's just kind of review that process really quickly. So just remember the key player here is going to be bradykinin, right? So bradykinin, and this is exactly what they're asking about here. They're really asking about this pathway. And this is why I tell you for step one, you got to understand the pathways. If you know the pathways, you can kind of rule in and out different things. You just have to make sure that you're solid on your mechanisms and your pathways. Okay, bradykinin is formed from high molecular weight kininogen. And then this bradykinin, just remember the functions of bradykinin, right? So some of the functions, one big one is vasodilation. So this can lead to edema, interstitial edema, right? As we start to vasodilate and we increase the permeability of capillaries and that sort of thing, that can definitely lead to edema. And with this vasodilation, like we said, there's increased, I'll just put perm here for permeability, right? This is all contributing to the edema. And sometimes we can also get pain, right, from bradykinin. So these patients can complain of painful edema, or in this case, this patient has abdominal pain, likely secondary to the elevations in bradykinin and this diffuse bowel wall edema. What is really regulating the step here is kallikrein. It's kind of stimulating the conversion of high molecular weight kininogen to bradykinin. Notice on here, when we're looking at this lab data, they also included, the C2 level, the C4 level, C1 inhibitor functions. Let's just talk about that too, because this is all kind of interrelated here. So also remember, and we talked about this a little bit in the video lessons if you've been following along, but remember that C2 and C4 can get converted to their subcomponents here, C2b, C2a, and C4a, and C4b. And this all happens by C1, right? C1 is going to allow for a lot of these processes to happen. Now, remember that there's a C1 inhibitor that regulates this process. So I'm just going to kind of draw this here. There's a C1 inhibitor that can regulate this process. So essentially, to put this all together, what happens here in this hereditary angioedema is that we have some dysfunction in the C1 inhibitor. Okay, so the C1 inhibitor has some kind of dysfunction. Now, there's different types of hereditary angioedema. A lot of this is beyond the scope of step one. In some cases, you can have low C1 inhibitor. I'll just put an I for inhibitor. But in all the cases, you have dysfunction of the C1 inhibitor. And so that's the thing that everyone should remember, right? So the C1 inhibitor is not going to be functional. It could be low if it's a certain form of hereditary angioedema. But in general, whether it's low or whether it's normal values, in both forms, the C1 inhibitor has some form of dysfunction. Now, you can imagine if the C1 inhibitor had some dysfunction, right? So if I knocked out the C1 inhibitor here, what's going to happen? Well, now this complement, the C1, is going to kind of be over stimulated, right? Because there's nothing to regulate it. There's nothing to keep it in check. And so you're going to get a lot of C2 and a lot of C4 getting converted because there's nothing inhibiting this C1, right? And so what happens is the levels of C2 are going to drop and the levels of C4 are going to drop because they're getting converted pretty rapidly here. And there's nothing to inhibit this process because we have a dysfunction or deficiency of this C1 inhibitor. That's the point. So with that being said, we know from this fact that we should have low levels of C2 and C4. Right? So if we're just kind of scanning this thing here, there's really only two options where we have low C2 or C4 levels, which actually is kind of nice. It simplifies this quite a bit. You can see that there is an option here where one is low and one is high, but again, we would expect both of them to be low in this situation, right? If we just kind of understand the mechanism. The other thing here is you can actually solve this problem just by knowing this and knowing that, hey, the C1 inhibitor function is going to be impaired, right? C1 inhibitor function is going to be impaired. We pretty much know that F is the right answer, right? Just based off of that information, we can solve this problem. But let's just explain why the kallikrein and the bradykinin are also elevated really quickly, okay? Because you may be in a situation in a board question where it's not quite this simple, right? You might have to literally go through every single arrow. So kallikrein and bradykinin are elevated. Why is that? Remember that the C1 inhibitor can also inhibit kallikrein, right? So if it inhibits kallikrein here, what's going to happen? So if kallikrein is inhibited, that's going to regulate this process, this conversion of high molecular weight kininogen to bradykinin. If I have a patient that has hereditary angioedema and they don't have C1 inhibitor, or they have a deficiency or dysfunction of the C1 inhibitor, then what happens? Well, if we don't have that C1 inhibitor functioning correctly, right, the kallikrein is going to kind of be overregulated, just like we saw down here. So the kallikrein is going to be more stimulated, right, because there's nothing to kind of keep it in check. There's nothing to inhibit it. And so we're going to get more high molecular weight kininogen converted to bradykinin, which means that we're going to have higher levels of bradykinin, right? And with higher levels of bradykinin, we get more vasodilation, more vascular permeability, more pain, more edema, which is exactly what we see here. Also, the last kind of layer here is to remember that ACE inhibitors run into this pathway also. So remember, this patient was given lisinopril, which is an ACE inhibitor. So to integrate ACE inhibitors here, remember that the ACE enzyme normally inhibits bradykinin. Okay, so the ACE enzyme, again, as a regulatory step, inhibits bradykinin. When you start somebody on an ACE inhibitor, right, you're going to inhibit this ACE enzyme. Okay, so if I start somebody on an ACE inhibitor like lisinopril, what's going to happen is I'm going to inhibit that ACE enzyme. If I inhibit that ACE enzyme, that's only going to further potentiate or stimulate this bradykinin, right? I'm going to get more and more of this bradykinin, or more functioning bradykinin, because I'm now inhibiting this enzyme. This was kind of like the last checkpoint. This patient at baseline probably didn't have a C1 inhibitor, so they already had kind of high levels of bradykinin, but now we added an ACE inhibitor and we actually made that problem even worse. And so again, the key is you can sit in a textbook and you can memorize, oh yeah, hereditary angioedema, so it's associated with edema, and ACE inhibitors make it worse. But the hard part is seeing that in a board question and just saying, hey, this is an ACE inhibitor, this patient has bowel wall edema, right? They have a family history, boom, you're thinking hereditary angioedema. Now you just have to understand the mechanisms to apply that knowledge. And so for those reasons that we just mentioned, again, the kallikrein should be elevated, right? We just said that. And the bradykinin should also certainly be elevated. So F here would be the best answer.
Hey everybody, Dr. Vizo here. In this video, we're going to talk about vaccinations. We're going to build a little bit on some of the other concepts that we already talked about with humoral response, cell-mediated immunity, and then how does that translate to some of these different vaccine types. So if you kind of know the rules of the game, if you're sharp on that B and T cell video lectures, then I think this is going to make a lot of sense. So before we get too far into it though, let's just go back to active versus passive immunity, and let's make sure that we really kind of nail this down. So when we're talking about active immunity, remember this is when we're going to have a host generating antibodies in an immune response. Okay, so the host is generating the antibodies, and it tends to be a more delayed humoral response. Remember, whenever you see humoral response, you want to think about antibodies in general. Okay, so we're thinking about plasma cells releasing antibodies. So in this situation, let's just say that this is our patient, right? This is our host. So in this patient in particular, it's going to be the patient or the host that is going to be producing the antibodies. Okay, so that's kind of the key with active immunity. And the thing to understand here is that this is going to take more time, right? It's going to take time to produce those antibodies because those B cells might have to undergo class switching, for example, or have to generate a memory response. But on repeated exposures, there's going to be a higher antibody response over time. This is really what we talked about when we looked at the IgG over time, right? We looked at a graph where we kind of said, okay, look, the first time that you're exposed, right? If this is your first exposure here, right? Your IgG response might be something like this. Okay, this is going to be IgG. But then later on, you might have a second exposure. Okay, so you might have a second exposure where you're going to have a lot larger IgG response, right? You might have a lot larger IgG response. But the thing that we also talked about when we looked at this graph is that both times we'll typically get an IgM response first, right, before we get the IgG response. And the IgM response shouldn't really change very much. Okay, so that was the important thing that we talked about when we looked at these graphs over time. But the point is that the antibody response in general, if we're just kind of looking at all of our antibodies, in general, that response should increase over time because we have active immunity, because we're generating memory cells. Okay, now when you're looking at each of these active and passive immunity, there's two kind of subcategories to be familiar with. And the two categories here are going to be natural and artificial. What kind of active immunity did we generate? You know, naturally, what would happen? How would I end up generating antibodies? Well, that's because I had an infection, right? So an infection would cause me to generate antibodies versus that pathogen. Okay, that would be natural. Artificial, on the other hand, would be if I perhaps was immunized, right? So if I was giving my patient a vaccination, I might be giving my patient some antigen potentially, or even a whole a virus, which we'll talk about in a second. But in any of these situations, it's still the host generating antibodies. That's going to take some time. time, but it was an artificial mechanism in which we generated that response. It wasn't naturally occurring, okay? We had to give that patient an immunization or vaccination. So in passive immunity, it's not necessarily the host producing antibodies. Instead, we're literally just giving our patient antibodies that have already been produced in some other fashion. Okay, so again, if this is my patient, so now it's not necessarily the patient that's producing antibodies, it's antibodies that are being given to the patient. Okay, so it's kind of the other way around. So you're thinking antibodies to the patient. These have already been preformed. It's not the host that's producing them. Okay, so that's why it's passive. It's right. When I think passive, I think of, there's not a lot of activity going on. Active, there is a lot of activity. It's the host producing antibodies. In passive immunity, we're just giving them, right? And so this is going to be very rapid humoral response. If I give you an antibody to an antigen, these antibodies can start binding. There's nothing to stop this antibody from binding to a pathogen right away. Right? Where with active immunity, it's going to take me some time to get repeated exposures to have really strong responses. Or it might even just take time in general to make certain antibodies like IgG. That's not the case. With preformed antibodies, I give them, they can start working right away. Okay, now the downside to this, though, is when we talked about active immunity, we made this huge response over time, right? We have a much larger IgG response on repeated exposures. The difference here is if I just give you antibodies, I'm not letting this host produce antibodies, right? I'm just giving them. Over time, these antibodies will eventually kind of wear down. Every antibody is going to have a half-life, right? In general, around 3 to 4 weeks, that antibody response is going to start to come down and it's going to decrease over time. And this is exactly what we see when we talk about mothers giving IgG to newborns, right? Or to the fetus potentially. Because remember, IgG can cross the placenta, whereas the other immunoglobulins are going to have some difficulty doing that, right? IgM way too big to be crossing the placenta, but IgG does in fact cross the placenta. And so this is an example of natural passive immunity, right? Mom's essentially going to be giving these antibodies through the placenta to the fetus, right? And that's exactly what's happening with natural passive immunity. It's a natural occurring mechanism that is allowing mom to give some of her antibodies from exposure she's had throughout her life to the fetus. But once that fetus is delivered, they're no longer going to be receiving those antibodies, right? Once this baby's delivered, now baby's not getting these preformed antibodies. Baby has some of them from mom from before, but over time, those antibodies are going to start to wear down. And that's where vaccinations will start to come into play, right, at certain times. Now, there's also artificial examples of passive immunity. So one of the artificial examples here would be giving a patient something like a tetanus immunoglobulin. So this is an antibody that's targeting a very specific pathogen, for example, or toxin, for example. And that's precisely what this tetanus immunoglobulin is. So you're giving the patient, again, antibodies into the serum that are targeting something very specific. Okay, so that's the tetanus immunoglobulin. And you'll see this for diseases like rabies, and we'll go into more detail on this, especially for step 2. But it's very similar in the sense that, again, we're giving a preformed antibody to a patient that has likely not been vaccinated or doesn't have these antibodies, presumably, to help fight off an infection or destroy a toxin potentially. Okay, so this is a big slide. So this is where we're going to talk about all of these different vaccine types. And the thing that you want to understand here is you want to know which type of vaccine falls into which category. And there's a lot of different mnemonics here on to try to understand, okay, well, where does varicella fall, or where does, the BCG or smallpox vaccine fall? And the thing that I want to call your attention to, though, when you start to go through some of these mnemonics, it's really easy to get the different vaccines confused. So what I like to do is I like to look at some of the examples where there's only 1 or 2 major examples to understand. And you'll see when you look at the live attenuated, there's a lot more examples just in general that tend to fall in this category. So I try not to necessarily, like if I'm just trying to memorize these, I try not to necessarily start here memorizing all of these. I'll start with something like my subunit, polysaccharide conjugated activated, and just know, okay, these are the big 3 here. This is the big polysaccharide, etc. And then, anything that doesn't fall in these kind of smaller example buckets, then you'll know that that's probably going to be in the live attenuated. But even what's more important than that is understanding the comparisons, right? So you have to know intranasal influenza versus intramuscular. You have to know Sabin versus Salk. These are the really big things that you absolutely want to be familiar with. And we'll talk about all of this as we go through it. But we're going to do this in building blocks. So I like having everything on one slide so you can just quickly compare it. But we'll do this in separate videos because it's a lot of material. So without further ado, let's start talking about the live attenuated vaccines. When we're talking about live attenuated vaccines, we're basically talking about taking a live pathogen, right? So if you want to use the example of a virus, okay, or you can say a bacteria. Let's just say a virus, okay, so I'm just going to draw some virus here. We're actually taking this virus, right, and we're putting this virus into our patient. Okay, so we're taking a virus and we're putting it into our patient. And you might say, well, that sounds really bad. We're basically just kind of infecting this patient. Now, the thing that's different though, if the virus normally uses, some factor to get into the cells, okay, so we'll just say this is its, this is related to its pathogenicity. Okay, so if it has something that's related to its pathogenicity that allows it to replicate, that allows it to infect cells potentially, what we're going to try to do is we're going to try to knock out the things that are really increasing the pathogenicity of this organism. Now, that's not to say that it can't replicate, right? They're live pathogens. They can still replicate. They're just less pathogenic, okay, and tend not to cause disease. But they're still going to have the antigens on their surface that we need to generate antibodies, right? So once we're given this live pathogen that just doesn't tend to be as pathogenic because we've made some modifications to it, then this patient will then have that live pathogen replicating, and then we'll produce antibodies against this pathogen. Now, the key thing here is, remember, you're adding in here a live pathogen. And what step one likes you to know is to know what kind of response are you going to generate. And this is where you really have to think about your pathways here. So just going back to what we've been talking about this whole time, right? So here's our dendritic cell. Okay, and we've gone through this a few times now. Now remember, a dendritic cell is going to have an MHC1 receptor on it, right? It's going to have an MHC1, and it's also going to have an MHC2. Okay, now most cells in our body, like we talked about, are going to have MHC1, most of the nucleated cells, if not all of them, but the MHC2 receptor is only going to be present on antigen-presenting cells. Since a dendritic cell is an antigen-presenting cell, it's going to have both of these receptors, right? Now, if I have some live pathogen that I just put in the body, a couple things can happen. The first thing that can happen is this dendritic cell can digest this pathogen. Okay, so you can have phagocytosis of this pathogen. Now, if that happens, the dendritic cell is going to present this pathogen that we just digested, it's going to present it on MHC2, right? It's going to present that pathogen or that antigen from the pathogen on MHC2. And we already know what this is going to do. MHC2 is going to trigger my T helper 1 cells, right? My CD4 positive cells, right? Rule of 8s. So CD4 positive cells are then going to bind to that MHC2 receptor using their T cell receptor and their CD4 positive co-receptor. And this whole process is going to lead to these T cells eventually activating B cells using CD40 ligand, right? We talked about all of this in the previous video. So if you're not familiar with this, go back, watch those videos. We go into this in great detail. And then we're going to have the CD40 ligand binding also. That's going to allow for class switching. And then these B cells will then become activated, right? And they'll go on to become plasma cells. Okay, so they'll go on to become plasma cells. And a plasma cell is essentially an antibody factory. And so now we're going to produce a bunch of antibodies to this antigen. Okay, that's pretty much the whole process. And that's going to be very consistent with what we see across the board. Now, remember what I said here. There's something really important. We're going to have CD40 ligand class switching occurring when this T cell uses this binding to a B cell. Okay, so remember, that's important. That causes class switching. Now, when you say class switching, what am I talking about when I say class switching? When I say class switching, I'm talking about taking IgM antibodies and turning them into a different heavy chain, right? So either IgG, IgA, IgE. Now, when we're talking about a memory response, we're primarily talking about IgG antibodies, especially in these simple kind of straightforward board questions. So when I say that there's T cells, right, these helper T cells go on to activate these B cells and they have class switching, what we would expect to see, right, is a strong immune response from this pathogen because we're going to generate IgG antibodies because we had class switching. That's an important piece to remember here, okay? So that's the first thing. You're going to get a good immune response because you're generating humoral IgG antibodies. You're going to have a memory response because you're involving T cells that are activating B cells, and those B cells are going to go on to have some memory B cells and also some memory T cells, and you're going to have these plasma cells pumping out IgG antibodies. The other part of this, let's go back a second. We said, okay, we went down this MHC2 pathway, and that MHC2 pathway allowed me to generate a very strong humoral response that involved class switching because we had T cell activation. What about if we go down the other road? Is it possible that this dendritic cell would present this antigen on MHC class 1? Okay, is it possible? Well, what would have to happen? Well, instead of the dendritic cell phagocytosing this pathogen, let's say this is a virus, right? Instead, maybe this virus infects this dendritic cell. Okay, so maybe instead of the dendritic cell phagocytosing it, the virus actually infects the dendritic cell. Now, if it does that, right, if this virus starts to put its viral material in this dendritic cell, we talked about this when we talked about these MHC receptors, this material is going to get broken down, put in the rough endoplasmic reticulum, and it's going to get sent up to this MHC1 receptor, okay? So now this is on MHC1. So what's MHC1 going to bind to? Well, if it's going to bind to anything, it's going to bind to cytotoxic CD8 positive killer T cells, right? So these again are T cells, but they're CD8 positive T cells. And again, the T cell receptor and the CD8 positive co-receptor are going to bind to this MHC1, and then this cell, realizing that there's something not normal here, right? Something that's not a self-antigen here, there's going to be perforins and other enzymes that are released to destroy this dendritic cell. And we're going to start to get some memory response from the T cells. Okay, so you're going to get a cellular mediated response. Remember, when you have these CD8 positive T cells destroying these dendritic cells or any cells in the body, that's going to be a cell mediated process. And so we say there's a cellular response or a cell mediated response and a humoral response. You can actually get both from a dendritic cell, right? Because the dendritic cell can phagocytose this, presented in MHC class 2, or the dendritic cell can actually get infected with this and then presented on MHC1 like a normal cell would. And because of this, we can get a cellular or cell-mediated response. Okay, so you can get a cellular response, and we can also get a humoral response. So we can get both because we gave this patient a vaccine that had a live pathogen. This patient shouldn't get sick, right, when they get a varicella vaccine or when they get a yellow fever vaccine, but they can get sick, right, because some of these pathogens can actually alter their virulence factors, right? This pathogen can alter some of these virulence factors, and it can actually then become pathogenic. Okay, so it can become pathogenic potentially, but that's not extremely likely. Now, where are we concerned about these pathogens potentially increasing their pathogenicity? We're primarily concerned about that in patients that are immunocompromised, right? Patients that don't have a good immune system are going to have a tough time responding to organisms that have acquisition of new virulence factors. Okay, now what patients are we going to be really concerned about in this category of immunocompromised states? Number one is infants. Now, most infants, and these are children younger than a year of age, most of them are not going to be getting these live attenuated vaccines. There's one exception to that, and that's the rotavirus vaccine. So the rotavirus vaccine is actually designed to be given usually around the first 6 weeks of life. Okay, there are some contraindications to the rotavirus vaccine, things like intussusception or history of GI tract malformations like a Meckel diverticulum. And this is more of a step 2 thing, but the reason for that is because you can have lymphoid hyperplasia in the Peyer's patches and you can end up with intussusception, right? Because this is generating an immune response, and so you can get lymphoid hyperplasia in infants. The point is, though, is that if you're going to remember, there's one exception to live attenuated vaccines. It's the rotavirus vaccine in infants. In the elderly population, we also will tend to start to avoid some of the more live attenuated vaccines. Pregnant females, same thing. These are kind of your big 3. And then always remember transplant patients that we're giving them immunosuppressive medications, or patients that have AIDS. Okay, so a patient that has HIV, this comes up pretty frequently. Now, let's say that this patient, you have to decide, hey, I have a patient here that has HIV. Do I give them their, varicella vaccine, for example, if they haven't had it? And how do you make that decision? Well, you have to look at the CD4 count. So if the CD4 count is greater than 200, right, which is usually the cutoff here, when we're talking about, an opportunistic pneumonia versus a normal pneumonia, we're going to look at the CD4 count, right? If you have a low CD4 count, you're significantly predisposed to opportunistic infections. If you have a normal CD4 count, even though you have HIV, it doesn't necessarily mean that you would have an opportunistic infection. The same is true here. You're not really considered to be extremely immunocompromised if you have a CD4 count greater than 200. However, if the CD4 count is less than 200, especially if it's significantly less than 200, these patients should not be getting live attenuated vaccines. However, the patients that have CD4 counts greater than 200, that's usually going to be okay to give them, especially in a board question. Okay, and the big vaccines here, like I said, you do have to commit some of these to memory. Varicella and MMR are probably the two highest yield, but you do frequently get questions where you have to distinguish different types of vaccines. So I would be familiar with the fact that the intranasal influenza vaccine is a live attenuated vaccine, whereas the intramuscular influenza vaccine is an inactivated or killed vaccine. In one case, you're giving them a live influenza strain. In the other case, it's a killed influenza strain. And we'll talk about that in a moment. And you're not really going to see intranasal influenza being given much in the United States. You'll primarily see the intramuscular variant. And then also remember that polio vaccines, there's two types. So you have Sabin and Salk. Okay, remember the Sabin is the live attenuated form, and the killed form is the Salk. And the way that I kind of remember that, okay, so the last letter is K, and these are going to be completely inactivated vaccines. And we'll talk about this in a minute. And so I remember that K for killed. So this is going to be a killed organism that we're giving the patient. And I always look at that last letter to distinguish the two. So K for killed. That's my easy way of remembering that. But you do have to distinguish these very frequently, so I would be familiar with them. And then BCG for tuberculosis, we'll talk about that probably in a future video. And then we also have smallpox here as well. But the big one here, I would say, especially for the US, know your varicella, MMR, rotavirus. It's going to be the exception in infants. And then for travelers, you'll see yellow fever occasionally. Not really going to give a whole lot of intranasal influenza in the US. Or the polio Sabin for that matter. And in the next video, we'll talk about some of the other variants here. All right, so moving right along, let's talk about the whole inactivated vaccines. And so this is going to be a non-replicating, as opposed to the live attenuated, non-pathogenic, again, as opposed to the live attenuated, virus, bacteria, or some type of pathogen potentially. And again, the idea here is that in the previous example, right, I gave this pathogen to this patient essentially. I just kind of decreased its pathogenicity in some way. In this situation, I'm going to just kill this organism and then give it to the patient. That's essentially the difference. So this organism cannot replicate. It cannot infect cells. And you might be saying, okay, well, how does that differ when you start to talk about some of these different pathways in the immune response? Well, let's think of it this way, right? If I just gave this patient a killed organism, can the dendritic cell still present a killed organism's antigens on MHC2? And the answer to that question is absolutely, right? This dendritic cell can still find this killed organism that's floating around there in the blood or the lymphoid tissue, and it can go up to it and it can phagocytose it, right? And so this dendritic cell can phagocytose this pathogen, and in doing so can present it on MHC2. And so that answers kind of the first question. If I can put it on MHC2, I know that I can get CD4 positive T cells to bind to that, right? And that can cause eventually B cell activation, CD40 ligand binding, class switching, and I can generate IgG antibodies to this pathogen. So I can generate humoral IgG antibodies to that pathogen, right? And that's what we also saw with live attenuated, right? We saw we could get a humoral response there with IgG antibodies, memory response, right? From the class switching from the CD40 ligand binding. The question here is, well, I don't see cellular on this side. Why is that? Well, let's think about this for a second. So again, if I kill this organism, okay, so remember now I'm killing it, can a killed organism infect a dendritic cell? Probably not, right? If this organism's killed, it's not going to be going around infecting dendritic cells. So can I present it on MHC1 on this dendritic cell? Probably not, right? Because it's not infecting the dendritic cell. For it to get onto MHC1, it's got to infect the dendritic cell. And in doing so, when a cell is infected, when any normal nucleated cell is infected, it's going to present that antigen, saying, hey, this is a virus inside of me, or this is a cancer. This is not normal. It's not a self-antigen. I need to get, basically, this cell is a suicide cell, or a cell that needs to self-destruct and destroy itself because that cell is infected. Now you have cytotoxic T cells that come in and destroy those cells. If this can infect a dendritic cell, it's not going to get presented on MHC1. So because of that, you're not going to get a memory cell-mediated response, which is going to decrease the efficacy / immune response that you're going to get from a killed vaccine. So that's why I put on here, you're going to get a stronger immune response in general from a live attenuated vaccine. Now, what's the upside to these killed vaccines then? Well, a big upside is you don't have to worry as much about acquisition of virulence factors causing higher pathogenicity because the organism's killed, right? We're not worrying about the organism getting stronger and causing an infection in an immunocompromised host. These organisms are killed, so it's very unlikely for that to happen. And the flu vaccine, which we're all probably somewhat familiar with at this point, and especially in the United States, you're going to see this given intramuscularly. And this is exactly what kind of vaccine this is, right? It's a killed inactivated vaccine. We're taking the influenza virus, we're killing in it, and then we're giving it to the patient. And that's essentially what we're looking at. And so they should generate a humoral IgG response. However, over time, some of that IgG, if we're not exposed to it, we might lose some of that memory response over time. We might lose some of those memory cells. And so for some of these vaccines, especially as we go down, you'll see there will be boosters that are involved. You might be saying, oh, is that why I get a influenza shot every year? Not quite. The reason you get an influenza shot every year is because proteins on the virus are kind of mutating or changing. And essentially, we're trying to predict every year what those proteins might be. And that's when that's what your flu shot is for. But in general, as we start to go this way, right, particularly when we get down to this polysaccharide inactivated vaccine, we're going to start to talk about why boosters become so important, okay? And you'll see hepatitis A, also whole inactivated. And again, knowing the differences here, right? Hepatitis A is a killed vaccine, but hepatitis B in particular is going to be a subunit protein that we're going to be giving the patient, particularly that Hep B surface antigen. And then again, we talked about the polio vaccine, right? So you have the Salk and the Sabin. Remember the K at the end for killed here, and then the Sabin is the active, and then also Salmonella typhi is in this conversation as well. But essentially, that's the big difference, right? The big difference is you're not getting a cellular response because the virus is killed. It's not infecting the dendritic cells to then have it be presented on MHC1. So that's the concept. So let's talk about these subunit protein inactivated vaccines. And so essentially what we're doing here is going back to our pathogen. We're saying, okay, look, we know that there's specific proteins on some of these pathogens that we can isolate. Instead of giving this patient the whole organism, let's just give them maybe a couple of those proteins. Okay, so maybe we just give them a couple of those proteins. And these proteins, we can then target these proteins and make antibodies against them. Okay, so we can take these antigens from this species, right, whatever it is that was on the cell surface, and we can make antibodies from that. All right, and so that's the concept. So we're just giving the protein components of the pathogen. So we wouldn't expect there to be any issues with, a replicating live pathogen or anything like that, because I'm just giving the proteins off of that virus's cell surface, for example, or from that bacteria. And again, you might be saying, okay, well, what kind of immune response are we going to generate here? When we can see it's humoral IgG. Why is that? Well, remember, if I have this antigen floating around in the blood, I can have a dendritic cell come up to that antigen. It can digest that antigen. Okay, so it can digest that antigen, and then that antigen can get displayed in MHC class 2 because I phagocytosed it with my antigen-presenting cell, and then eventually lead to the formation of antibodies, IgG antibodies from class switching. It's the same process we keep talking about. And again, you might say, well, was there a cell-mediated response from this? And the answer is no, right? There's not going to be one for the same reason we talked about when we talked about the whole inactivated vaccines, because this antigen is not a live organism infecting cells, right? It's not going to infect a dendritic cell. It's just a protein component floating around in the serum that we're generating antibodies to and phagocytosing. Okay, so for that reason, you're not going to get any cell-mediated response, again, with a subunit protein inactivated vaccine, right? You're just giving this patient a protein or an antigen from that pathogen. So you're only going to get humoral IgG response. If we're stratifying this is going to be in the middle somewhere in terms of its immune response. Pertussis is very classic. And then we also have hepatitis B, right? We talked about that a little bit in the last video. Hep B surface antigen, and we'll talk about this in greater detail when we talk about the microbiology section, but the Hep B surface antigen is going to be what we're targeting. So again, it's just like it says, right? If I have some virus here, if this is my Hep B virus, okay, I'll just put HB here for my Hep B virus, and on that Hep B virus, there's going to be certain surface antigens, okay? And instead of giving the patient a vaccine that has the whole Hep B virus, I might just give them the antigen, right? I might just give them what the antibody targets, for example. Okay, and then we can also see HPV is on here, and then toxoid vaccines. And what we're doing is we're essentially giving the patient this toxin, but its pathogenicity has been wiped, right? So it's not going to be causing disease, but it still has immunogenicity. Okay, so I can still make antibodies to it. So pathogenicity down, immunogenicity, we can still generate antibodies to, and that's the concept. Okay, so that's what we'll see with tetanus and diphtheria. And sometimes you'll see this vaccine, right? This is going to be a very classic vaccine, especially when you get to step 2. You'll see the Tdap vaccine, right? Tdap. So what does this stand for? So the T is for tetanus, right? That we just saw. Okay, so that uses a toxoid. Then we saw the D here. This is for diphtheria. Okay, and what is this AP? AP is what we have right here, right? AP is acellular pertussis. Acellular pertussis. Why does it say acellular? Because the organism is not being given. We're just giving the subunit protein with 2 toxoids. Let's start talking about the subunit polysaccharide inactivated vaccines. How does this polysaccharide differ from the protein variant? Well, to answer that question, let's start with a bacteria here. So we're just going to take some bacteria, and maybe this bacteria has a capsule. Okay, maybe this bacteria has a capsule. All right, and let's just say that this is a polysaccharide capsule. When I say polysaccharide, what I'm basically saying is that the capsule around the bacteria is made up of carbohydrates. It's not made up of proteins. Remember, proteins are peptides. Polysaccharides are carbohydrates. Okay, these are different substances, different macromolecules. So what I'm saying here is, if I'm thinking about making a vaccine for this bacteria, do I want to make a vaccine where I give the patient the whole bacteria? Maybe, right? That might be, an option, giving them a live attenuated vaccine, but maybe I don't want to give them the whole bacteria, right? I don't want to worry about this thing potentially infecting my patient. So I just say, look, I'm just going to give them the polysaccharide. Okay, so if I just give this patient part of this polysaccharide capsule, maybe that can generate an immune response to produce some antibodies. Okay, so let's think about this. Can the dendritic cell see this polysaccharide as an antigen and then present it on MHC2? Well, here's the problem, right? A dendritic cell that may recognize this polysaccharide, for example, it's not going to be able to put it on MHC2. And you might be saying, well, why is that? Why can't it just display it there? Because remember, MHC only displays protein antigens. Okay, that's something we talked about. It only displays protein antigens. This is not a protein. This is a polysaccharide. So I cannot display this on MHC2, which means if I cannot display this on MHC2, I cannot activate the T cells. I can't generate a memory T cell response, which means I can't then cause class switching to occur, which then means that I will only have IgM antibodies responding to this polysaccharide capsule. Now, in real life, it's a little bit more complicated than that. But remember, if you have B cells around that recognize this polysaccharide capsule, they can still make IgM antibodies, right? They can still do that. However, you might get an inflammatory response potentially, but it's not going to be a strong one. It's not going to be a super strong memory response because if there's only polysaccharides around in this capsule, then this is going to be a T helper independent response. And we talked about this. This is only going to generate a humoral IgM response, not the IgG responses that we generated with all of these other vaccines. So this is going to be the weakest immune response that we've seen thus far among our vaccines, just from a general perspective. And the classic example of this is going to be the strep pneumo vaccine PPSV23. And this vaccine is also sometimes called Pneumovax. Okay, so this is sometimes called Pneumovax. And the reason that it's given, you might be saying, well, why do we even give this thing at all if it's so weak? Well, it covers a lot of different variants and strains of strep pneumo, okay, 23 of them to be specific here. And so that's why Pneumovax is sometimes given. There's 23 different serotypes that we can target. But the big thing is you start to get levels that decline after 4 to 5 years, depending on how robust your immune response is. And so you need very frequent boosters. And so that's kind of the catch. Because you're generating this IgM response only, you're going to need even more frequent boosters, even more frequent than you might need for some of these other vaccines. Okay, so that's something to keep in mind. There is a way kind of around this for certain vaccines where what we can do is we can take this polysaccharide and we can say, look, I can maybe attach this polysaccharide to something that might be able to get through this pathway. I might be able to attach this polysaccharide to a protein. Okay, because if I can attach this polysaccharide to a protein, then I can get this whole thing potentially presented to an MHC2 receptor. Okay, because then we have a protein component with it, right? And this thing here is essentially going to be my protein. Okay, so the protein, I can pick any protein that I can bind this polysaccharide to, and this whole thing together is going to be called a conjugate. Okay, so this whole thing, when I combine these two, this is going to be called my conjugate. Okay, so I'm essentially conjugating, I guess you could say, a protein to a polysaccharide. Okay, so that's essentially what we see down here, the polysaccharide plus the protein. And the idea is, now that I have this protein and this polysaccharide here, I can put that on my MHC2 because my MHC2 can certainly display the protein. Okay, and then I have my polysaccharide here. And now these T cells can essentially bind to this MHC2 and we can still generate antibodies with class switching, right? Because now I can go down this pathway and I can get IgG antibodies. So I can get a humoral IgG antibody because I have a protein component now that this dendritic cell can display. And again, will I generate a cellular immune response? The answer is no, right? This thing, which is part of a bacterial wall attached to a protein, dendritic cells will phagocytose it, but the dendritic cells will not be infected buy it per se. And so the classic example of the vaccine here is going to be the PCV13 vaccine. And the PCV13 vaccine, this is sometimes known as the Prevnar vaccine. You know, the easy way to understand these vaccines that fall into the polysaccharide / conjugate category is just remember, if you have a patient and they have a splenectomy, what are the organisms you vaccinate for, right? It's these organisms because they all have capsules, polysaccharide capsules. Okay, so you can see strep pneumo, meningococcal vaccine, right? Haemophilus influenza type B, right? And again, strep pneumo for patients that don't have spleens. And we already kind of talked about that, so I won't go into too much detail on that. The last thing I want to talk about here, just really briefly, just be familiar with, you know, some of the RNA vaccines, specifically the mRNA vaccines. We're essentially taking, you know, a strip of code, putting that in our host. Okay, so let me kind of illustrate this here. So instead of putting in a polysaccharide or a live bacteria or virus, what we're doing is we're putting in a strip of code. So I'm just going to kind of put this out here. So this is just some strip of mRNA. Okay, and so this strip of mRNA is going to have a specific code on it. And we're going to put that in the host. The host will have cells that take up that mRNA. And our cells will then generate specific proteins from that mRNA. Now, in the example of COVID-19, quite simply, we'll generate spike proteins. Okay, so we're making, our own cells are making the spike protein from this code of mRNA that was given to the host, right? And our own cells take up that mRNA and we start translating that mRNA and start making these proteins here. So we start to make these spike proteins, and then we make the spike proteins, right? Our own cells make them, and then we make antibodies to the spike proteins that our own cells made. So that, I know it's kind of confusing, but again, this is not super high yield at the time of this recording, although I do think that it will start to become asked about a bit more frequently, as this is certainly a global phenomenon right now. And again, the classic example for this would be the SARS-CoV-2 virus or COVID-19. The last thing I want to say is that for some of these vaccines, especially the polysaccharide vaccines, you can give things like adjuvants. Okay, so you can give things like adjuvants. And an adjuvant, essentially what this is it's something that will allow for there to be a higher, stronger, more robust immune response. And we don't have to go into a ton of the details on this because it's kind of in that medium low yield rating. I just thought I'd bring it up. But the big one to be familiar with when you're talking about adjuvants is aluminum. Okay, so aluminum is the one, if you're going to remember any, you give the patient aluminum inside of that vaccine or as a component of that vaccine, it can tend to boost the immune response under specific situations. So just something to be familiar with. But hopefully now you have a little bit of an understanding of these different vaccines. And I always just go back to the simple pathway. If you're ever in doubt, right, is it a protein? If it's a protein, you know it's going to go on MHC2, right? If it's not a protein, it's not going to go on MHC2. If it doesn't go on MHC2, you're not going to get class switching, right? That's the concept. If it's live and it's infecting cells, you're also going to get a cellular response. Because if this dendritic cell gets infected, CD8 positive T cells will also be activated. So that's the concept. Remember which vaccine goes where. Remember polysaccharide, just think, what are the things you vaccinate against if you have a patient that has a splenectomy? Those are the vaccines we're talking about. And then I think you can pretty much, I just remember the subunit protein here as pretty much Tdap with hepatitis B in there. And then the whole inactivated versus the live attenuated. I think a lot of these make a lot of sense because we don't give live attenuated influenza vaccines. And then I already talked about identifying polio, right? Remember the K in Salk for killed vaccine or whole inactivated versus Sabin, right? And so I think some of these will make a lot of sense once you go through this once or twice.
Take 15 seconds to pause the video, read the question, and then we will go through the answer. Okay, so we have a research group attempting to identify the effectiveness of SSVK, a vaccination against Shigella sonnei bacteria. All right, isolated Shigella sonnei were grown in broth containing deoxycholate. Formalin was added to the vaccination sample for complete inactivation of pathogenicity. In preclinical trials, the vaccine demonstrated immunogenicity against various strains of Shigella sonnei infected mice. Okay, great. Response per participant was recorded as strong or poor using cutoff serum values of specific immunoglobulins. Data collected is shown below, which the following most likely describes the SSVK vaccine. Okay, so I think we're trying to figure out what type of vaccine this is. There's definitely some clues here off the bat. Just from reading this line, there was complete inactivation of pathogenicity. So remember, there's two different terms being thrown around here. There's pathogenicity and then there's immunogenicity. You got to kind of keep these things straight, right? So pathogenicity, remember when we talk about pathogenicity, primarily talking about, the infectivity of this, organism or of the vaccine, right? So if I give somebody like a live attenuated vaccine, it's not a very pathogenic vaccine, but it still can become more pathogenic, right? There could be spontaneous mutations or alterations that can make it more pathogenic, which is why, if you give a patient, especially an immunocompromised patient, a live attenuated vaccine, these patients are at higher risk of developing an infection, right, from that vaccine. So the point is you can have some, inactivation of pathogenicity, but the fact that they're adding this formalin and there's complete inactivation of pathogenicity already kind of takes me kind of out of the realm of the live attenuated. Now remember, this is different though than immunogenicity. It's referring to our ability to generate an immune response, right, from that vaccine or from that antigen, whatever it is. So we can have a really high immunogenicity. We can generate a really strong immune response from something that's not very pathogenic. Conversely, right, you can have a pathogen that has really high pathogenicity. It can cause really harmful disease, but it's not very immunogenic. It's difficult to destroy, right? So, or at least difficult to generate an immune response against. We're looking at specific immunoglobulins, right? So they have the specific idiotypes to SSVK here. So let's see if we see anything significant. So they said that response per participant was recorded as strong or poor, which is what we see here, using cutoff serum values of specific immunoglobulins. And I can see that all the participants here were accounted for. So there's 73 participants in each of these specific diagnostic buckets here. So again, SSVK specific serum IgG. Okay, so it looks like overall, these patients that were inoculated developed a pretty strong response, generally speaking. A couple people did not. Serum IgA, same thing. Pretty strong response throughout. Fecal IgA, again, pretty strong response. So this is significant because we know that this vaccine allows for class switching, right? So that's the significance of these, because in each of these situations, right, we would need class switching to occur. Okay, we would need class switching to get this robust response of immunoglobulins that are not IgM. SSVK specific CD8 positive cytotoxic T lymphocyte assay. Okay, so they're telling us these are essentially looking at the amount of CD8 positive T cells that are responding to this antigen, I suppose. And the strong response here is 0. So this is interesting. So, this is basically telling us that, look, we have humoral immunity, right? So we have humoral immunity. Okay, so we have humoral immunity. And we know that the antigens are T helper dependent antigens. And you might be saying, where are you getting all this information from? Okay, I'll kind of walk you through this in a second. We talked a lot about this, by the way, in the vaccine videos, if you haven't watched those already. But the thing that I know that that's not happening here is we don't have cellular immunity. Okay, we don't have cellular immunity. And how do I know that? The fact that this CD8 positive assay to SSVK specific CD8 positive T cells, we pretty much don't have any T cells that are responding with cellular immunity to this antigen. What does that mean? That means that this antigen from Shigella sonnei is not invading our host cells, right? It's not invading antigen presenting cells. Remember, if I have an antigen presenting cell here, right, just going back to the basics, and again, we go into way more detail in the vaccine video, but all of the nucleated cells in the body should have an MHC1 receptor. If I had a virus that was invading a host cell, right? Let's just say it invaded, epithelial cells, invaded, tissue throughout the body, but it also can invade antigen-presenting cells, right? This virus can essentially get into this antigen-presenting cell, and then these viral particles that we end up producing in our host tissue can get presented on MHC1. And these MHC1 receptors containing the antigen can then present this to a CD8 positive T cell, and this whole thing can go on to produce cellular immunity, right? Eventually, what we probably do, we would probably end up destroying this antigen presenting cell using perforins and granzymes and that sort of thing. But the key thing that you had to understand here is that we had to either infect this antigen presenting cell using a live strain of bacteria, right, that had some pathogenicity, right? It has to have some pathogenicity to do this. Or the other option is I could have given this patient a DNA or an RNA sequence, and instead of there being a virus that infected the cell, I could have just given this patient some RNA or DNA that goes into the antigen presenting cell and then codes for all of these different receptors, right? Or all of these different antigens that then get presented to MHC1 and then goes on to activate a CD8 positive T cell. And again, this is not just happening in antigen presenting cells. I'm just using antigen presenting cells so I can juxtapose it to humoral immunity. But this can happen in any cell, right? Because all nucleated cells should have MHC1 receptor. So the point is, again, I either had to infect this cell right, with an intracellular pathogen, or I had to inject DNA or RNA so that the host cell can produce viral particles that get presented on MHC1 receptors to CD8 positive T cells. I can tell you off the bat, just reading this line here, complete inactivation of pathogenicity, that if there's complete inactivation of pathogenicity, then we know we're not going to get cellular immunity. This is probably not a live attenuated vaccine just for that reason. If I look at this specific CD8 positive cytotoxic T lymphocyte assay, I can see there's no response really at all here. In general, there's a poor response. So I would assume that there's not very good cellular immunity. Now, live attenuated vaccines, the big difference between a live attenuated, a whole inactivated, and a subunit polysaccharide is the live attenuated has cellular immunity. And I know that there's probably not any cellular immunity going on, at least that's what's suggested in multiple ways by the question. So it's probably not live attenuated. DNA sequence vaccines are not very high yield in general. Again, this involves kind of injecting DNA in so that we can produce an antigen. Same thing with RNA sequences. We talked a little bit about them in the vaccine video, but essentially it involves kind of like injecting the code to produce the antigen by our own cells, which is what you see with some of the early, novel coronavirus vaccines. But in both these situations, because we're producing these antigens, they can get by host tissue, they can get displayed on MHC1. And so these can induce cellular immunity. Okay, now this is another situation where you might not have necessarily known that specific fact, but there are other clues in the question to kind of get you to a more reasonable answer, which is the whole inactivated vaccine. Okay, so the key thing here to understand between the whole inactivated and the subunit polysaccharide is both of these can allow for humoral immunity. So essentially what's happening here, again, we're going to take our same kind of antigen presenting cell here. The difference here is, right, so these antigen presenting cells, they're going to have the MHC1 receptor, just like the MHC2 receptor, right? So they have their MHC1 receptor. I'll just put one here, and they're going to have their MHC2 receptor. Okay, so this MHC2 receptor is going to basically allow us to present extracellular pathogens or antigens from those pathogens. Because what happens is, right, if I have a bacteria like Shigella, right, if I have this bacteria around and I phagocytose this bacteria, now I can take components of this bacteria like an antigen and display it on an MHC2. The MHC2, right, 2 times 4 is 8, right, rule of 8s. This can bind to a CD4 positive T cell, and then this can stimulate humoral immunity. And you might be saying, well, can't you do that with live attenuated vaccines too? Yeah, absolutely. You can still go through that same process with a live attenuated vaccine also. This is why live attenuated vaccines will allow for cellular immunity and humoral immunity, right? And we go through like all this, like I said, in the vaccine video. But the whole inactivated vaccines, these don't allow for cellular immunity because the pathogen is dead, right? We've completely inactivated it. They said here, they put it in formalin, it's completely inactivated, and you can see that there's no cellular immune response. So a whole inactivated vaccine does not have cellular immunity. It does have humoral immunity though. It has humoral immunity because we can still produce antibodies to this. This antigen presenting cell can still present this to CD4 positive T cells, right? And we can eventually undergo class switching as this binds to a B cell, right? CD40 ligand, and that can allow for humoral immunity, right? Antibodies being generated. Now you might be saying, well, what's the difference between the subunit polysaccharide vaccine?